Thanks all for coming to Charles Song Award Ceremony and Distinguished Lecture. I'm Peter Kang, Assistant Professor in Earth and Environmental Sciences. And I'm here to introduce and say a few comments about Charles Song Fellowship and introduce Professor Judy Yang for announcing this year's award winner. So Lian is traveling now, and I was very happily agreed to introduce Song Fellowship on his behalf. And 2 years ago, we lost an esteemed colleague, dedicated teacher and scholar, and internationally renowned scientist, Professor Charles Song. Even though I never personally met him and he no longer with us, I'm very aware that Professor Song's legacies of excellence, kindness, and caring are alive and we're here in the lab and across the world in our software network and alumni and friends. So last year, a generous group of his former students initiated Charles Song Fellowship at SAFO to honor Professor Song for his pioneering research and especially his caring mentorship. So it has special meaning, and the new fellowship provides graduate student support, especially for international students at SAFO. So I want to share just 2 quotes from his former students. The first one is, a young person getting started in the world needs a mentor, someone that takes an interest in them both professionally and personally with encouragement and guidance. Professor Song filled that role in my life. The second quote says, Professor Song himself was an immigrant. He understood the challenges that international students faced abroad. He always reached out to new international students and offered his help and support. As I said, even though I never met Dr. Song, it was not hard to imagine how great and inspiring mentor he was and scientist. I think SAFO is really fortunate to continue celebrating Dr. Song's legacy through this fellowship. And while preparing for this opening statement, I myself got inspired and made me to look back and think about what a good mentor is for students. So with this, I'm very happy to ask Professor Judy Yang to introduce this year's Song Fellowship winner, and Judy is his advisor.
Thank you, Peter. It's my great honor to be here to introduce the winner of the Charles Song Fellowship, Shih-Hsun Huan. Shih-Hsun is currently a student at Civil Environmental Geoengineering and St. Anthony Falls Laboratory. So Shih-Hsun got his bachelor and master degree in civil engineering in National Taiwan University. He's a top student with top GPA, and he has done very excellent job on numerical simulation of suspended sediment transport. He joined my group in 2020, which, as many of you know, is the most difficult year to start a PhD career. Like the lab was closed, we have to apply to go back to the lab, and there was a social distance, and the kitchen was closed, so there was no place to eat. And Shih-Hsun was my first student and the only student then, so he was very lonely. I was very concerned about Shih-Hsun, to be honest, because his background is in numerical simulation. He didn't do any experiments. And as many of we know, most of the experiments failed. So at the beginning, I remember there was one time Shih-Hsun came to my lab feeling very frustrated. It was like, everything's not working. So I was very concerned, but it turns out my concern was not necessary. Shih-Hsun quickly adjusted, and he was able to design new sets of experiments that I have never done before. And he was able to analyze the data and find new discoveries and publish in top journals. And over the past 3 years, almost 3 years, 2.5, I found Shih-Hsun, you have become more optimistic, more resilient, with more grit, and I'm very proud of you. You are a very important member of my group, and you are always organizing events, helping other lab members, and a very responsible TA. And I believe the spirit, the grit, and the resilience is as equally important as the academic skills you get. And I believe the spirit will help you succeed, overcome all the challenges in your PhD and in your future career. And I'm very glad and Please come to accept the award. Congratulations, Peter. Congratulations, Shih-Hsun, and Peter is going to announce the award.
After his talk.
After?
Yeah.
Oh,
Yeah.
sorry, my fault. You can present, Shih-Hsun.
Yeah, maybe you can first present your short talk and then we will present the award.
So, hello everybody, I'm Shih-Hsun Huan, and it is a very great honor for me to be here. And I'm a PhD candidate of the Department of Civil, Environmental, and Geoengineering, and my lab is in the Safo. So sometimes I look at the Mississippi River beside Safo. I was wondering what I will see if I'm a water molecule that's traveling to downstream. Will I spend months in the lake or just going to the sea? Actually, sometimes water molecule will make a little detour into the sediment bed and then comes out of it after some times. And we call this phenomena hyporheic flow. And it will happen especially when there was vegetation and large wood gems in the middle of the channel. So today I will talk about how I design experiments to investigate the hyporheic flows induced by vegetation and large wood gems. So first of all, let's talk about what is the hyporheic flow in the hyporheic zone. In the channel or in the river, there was a region that the surface wall will actively exchange with the pore fluids in the sediment. And this region we call it hyporheic zone. And in the hyporheic zone, there was a kind of flow that will run into a sediment bed and also sediment bed after some space and time. And we call this hyporheic flow, hyporheic flow. And because the flow runs in and out of the sediment bed, so it will carry the gases, the pollutants, and nutrients into a sediment bed. And it also gets the organic waste out of sediment bed. So apparently, it controls the pollutant transports and biogeochemical cycles in the sediment bed. And thus, it has a great impact on the biodiversity of the aquatic ecosystem. You may ask, what kind of factors that will induce such kind of flow? In fact, any factor that can generate the pressure gradients along stream beds can generate hyporheic flow. For example, the backflow, the standing wave, even the turbulence itself. And among all the factors, there are two factors getting more and more attention this year. The internal vegetation and large wood debris. In the field work, they already showed us the presence of these two factors in the channels will induce hyporheic flow. However, how these two factors impact the hyporheic flow have not been fully understood because they are not transparent, especially the sediment itself. So as I say, the hyporheic flow is the flow that's run in and out of sediment bed. So basically this process happens within the sediments. If the sediments are not transparent, so it is very hard for us to see what exactly happens within the sediment bed. So to address these problems, I'm using a new material, hydrogen beads. Actually, hydrogen beads is kind of gel that's a mass of polysaccharide. So theoretically, it's edible. So you can imagine there was a box of bubble tea in the picture. Although there are some bubble tea, oh no, hydrogen beads in the box, we can still see through the box to see the black and white panels behind the box. So because it's transparent, if I'm using this as my sediments in the lab, then I can study the hyporheic exchange or hyporheic flow. For example, actually in the box, as these pictures are filled with hydrogen beads. So when I inject the dye into this box, I can see exactly where the dye goes. And if there is hyporheic flow that can carry the dye, then I can check the hyporheic flow. So this is the back concept of my experiment. And following, I will talk about how I use this kind of design to investigate the hypertrophic flow induced by in-channel vegetation. So this is the flume I work with. This is the eco flume in the sample, and it is a water recirculating flume. And I use it to simulate the river. And in text sections, I fill the sediment bed part with the hydrogel beads, and I instill some aquatic dots to simulate the vegetation. So what I do next is I will inject dye into a sediment bed and see how the dye be transposed by the hypertrophic flow. And in addition, because the dye I use is fluorescent dye, so when I shine the dye with blue light, the dye itself will emit green light, and the light intensity emitted by the dye is proportional to its concentration. So it not only means I can know where the dye goes, I can also know how much of dye is at a certain location. So this is what happens in the experiments. In this experiment, as I said, I instilled several dots to simulate the internal vegetation. And the black pattern is the meshes I used to host the hydrogel bead. So the hydrogel bead doesn't move. And in the channel, I uniformly inject the fluorescent dye into the sediment bed and run the water. So you can see that the dye was gradually released out of sediment bed by the hypertrophic flow. And to quantify this process, I'm using a, okay, so if you look closely, then you can see that most of the dye was transposed out of sediment bed as the back of the dots. So definitely there were some interactions between the dots and the hypertrophic flow in the sediment bed. So to quantify this process, I'm using a downward looking camera. In this video, this is a top view of the channel. So the flow is from the bottom to the top, and the blue dot is the top of the dots. The black patterns is the meshes, and the green region is the locations that I inject the dye. So you can see that when the time goes by, the fluorescent intensity detected by this camera decrease. That means more and more dye be released out of the sediment bed. And if we plot the fluorescent intensity with the time, we can get a curve. We call it washout curve because it tells us how quick the dye be released from the sediment bed or wash out from the sediment bed. And here is two samples. The first curve, the black curve, is for the case without vegetation, and the red curve is for the case with vegetation, in the channel with vegetation. So we can see, and these two cases are run on the same mean flow velocity. So we can see that with the same mean flow velocity, the dye in the channel with vegetation, which is red curve, is washed out quicker than the dye in the channel without vegetation. So we can say that the percent of vegetation increase the hypertrophic flow. And to compare these two curves quantitatively, we're just using a simple exchange model, which can simulate the exchange between the surface flow and subsurface region. And in this model, there was a parameters called hypertrophic exchange velocity. And by fitting these models with the curve, we can get a value VH for each case. And we compare the hypertrophic exchange velocity between different cases. So this is the results. In these pictures, the x-axis is the mean flow velocity within a channel. The y-axis is the fitted hypertrophic exchange velocity. So again, the blue mark, the black mark is for the case without vegetation, and the red marks is for the case with vegetation. So we can see that as the same mean flow velocity, the presence of vegetation can increase the hypertrophic exchange velocity by a factor of 4. However, these pictures can also tell us that if we want to predict the hypertrophic flow or hypertrophic exchange within a channel with vegetation, we cannot rely on the mean flow velocity. because with different setting of the vegetation, the hyporheic the behavior of the hyporheic exchange velocity is different with the mean flow velocity. And we saw of that the presence of vegetation in the channel is not only increase the resistance of surface flow in the channel, but also they will create the turbulence. So we think maybe if we evaluate how turbulence a flow is, we can see the impact of vegetation on the surface flow. So here we measured properties called turbulent kinetic energy, which basically means how turbulent a flow is. And we plot the VH with the turbulent kinetic energy, and we find that both data sets collide with each other. So that means we may using the turbulent kinetic energy to predict the hyporheic exchange in a channel with vegetation. So in these studies, I designed experiments to quantify the hyporheic flow induced by vegetation. And I also proposed a turbulent-based model or theory to predict the hyporheic exchange. And the above contents has been published in the geophysical research letters last year. And for this year, what I'm working on is the impact of the log jams on the hyporheic flow. So I'm using the same flow, but this time I didn't install the vegetation tiles. I installed the model of log jams and run the flow. And in the videos, you can clearly see the hyporheic flow path caused by the log jams. And the next step I'm working on now is to quantify this process. So thank you. This is my study. And my study is funded by the NSF and Minnesota Water Research Fund. And I want to thank my good mates. They not only support my study, but also support my life. I'm not lonely anymore. So I also want to thank the engineering staff, the band and mans, and all the students that helped me within this experiment. So thank you.
Any questions to Shih-Hsun? I have one brief short question. Can you comment on the effect of porosity? So you used hydrogel, which I guess porosity is about maybe 50%?
It's about 0.3.
0.3. So as you decrease porosity, do you think this turbulent kinetic energy can still predictor of the hyporheic exchange, or do you think you have to add somehow the porosity factor?
So because in the models we use to evaluate the hyporheic exchange velocity already consider the porosity, so I think it should work. Yeah.
Any other question? Yes, Jeff.
Do you have any thoughts on why?
Do you want to use the microphone? Yeah.
On the plot before, you had one of your data points wasn't perfectly on your turbulence kinetic energy relationship. Do you have any thoughts why that deviation, the red one to the right,
So
on the right plot?
you mean this?
Yeah.
This
Do you have any thoughts why?
Because I think it's when the flow velocity increase, the vegetation itself will just like eliminate the transport of turbulence and the energy. So maybe when there was not much of the energy can convert to driven the hyporheic flow anymore because the limitation of the tiles. Yeah.
Okay, if there's no more question, maybe Judy, you can come up and we can present the work together to Shih-Hsun. And I think we will have official photo with Mike after his talk. Yeah. So Shih-Hsun, congratulations for a very well deserved award.
Thank you.
Maybe we will see.
Yeah.